We went to Baylor College of Medicine, which is in Texas Children's Hospital at Houston. We spent 2 weeks there understanding the protocol for SARS-CoV-2 sequencing.
Melendez received hands-on training.
So sequencing is basically a method, testing method that we use to identify the different nucleotide pairs of which makes DNA. And based on DNA, that's everybody has a unique code. So based on those code, we understand that the virus is SARS-CoV-2. The DNA is different from somebody else, and we can use it in different departments such as forensic, and also in the medical field and research, and also in culture, cultivation of crops.
In this case, Melendez will focus his efforts on detecting COVID variants. This nanopore sequencing machine plays an integral role in the process. Linked to a CPU, it generates the data from different strains necessary to run against a global database of variants already detected.
Medical field speaking, the research doesn't necessarily will generate any data for the physicians to create a treatment for it, but rather just to enhance surveillance in Belize. Sequencing is not a day procedure, and it's not cheap also. It takes approximately 3 days to complete a sequencing for 44 samples, approximately. And it takes up 3 days, as I mentioned, and during those 3 days, there are different procedures that need to be taken into consideration in order for you to have a good sequencing results. Nanopore technology has different kits that can be used. The kit that we're implementing can only be used approximately, has a lifespan of 4 runs. So within those kits, those are also costs. We'll put a load on the budgeting.
The central lab currently has 5 of these kits on hand.
So the aim, I think, is the acting director of the laboratory is creating a algorithm, because obviously we can't sequence the entire population of Belizeans. So we'll look at different age groups. We'll look at different location, geographical location, where are. So all those criteria will be met in order for you to have a good sequencing outcome.
And obviously, I suspect that when we're doing our sequencing, we're not looking for any new variant. We're mostly seeking to detect variants that already exist, correct?
That is right. But also, since the virus is mutating rapidly, and I mentioned that the data generated, it's compared to the database. Okay. So that database will have, if there is a new variant coming out, then it will also go ahead and detect that variant.
Melendez says while there are only 2 persons trained at the lab for this, once the equipment is up and running, additional members of staff will be trained. Reporting for News 5, I am Paul Lopez.